All right. So, hi everybody. Plan for today is to continue talking about cellular innate immunity. As always, I got overly excited when putting together these slides, so I've got a bunch of random stuff in here. We'll see what I get through. It's really cool stuff. It's just a timing question. I also had a very unfortunate realization last night, which is that apparently my videos that I've been posting to YouTube have not been posting to YouTube. They've been going somewhere in the ether when I hit post. Because I tried, last night I was posting the last one, and I was like, oh, I don't feel like remembering the setting for this. Let me copy it off of one of the other ones. And so I tried to open one of the old ones, and the old ones weren't there. And this is at like 2 o'clock in the morning, and you can imagine that I was really excited, and feeling very positive about everything at that moment. I think when I was just over in my office, I at least did what the internet says is supposed to fix it, but I have not actually tried to re-upload anything to see if that fixes it. So they're coming. That's all I know. Some of them are going to be easy fixes. Some of them are going to be hard fixes. So we'll see what's going on. That's the story with that. You all have your lab stuff back, and we will deal with what's going on in lab on Wednesday. Also remember that you have the PRR assignment due on Friday, where you've got the 2 papers that you're supposed to read, and then there's some questions that you want to answer about those. I am going to mention a few things sort of related to that today, so that might give you some other info. But to be perfectly honest with you, there are 2 papers. You have to kind of say what's important about each of them, and then which one you think proved stuff to you more. I legitimately want to know what you think. I wouldn't have made this assignment if I felt like there wasn't a good argument to be made for both of them. And I'm legitimately interested to hear what you think. So don't feel like you're trying to read my mind of what the right answer is or something. The right answer is I legit want to know what you think. Yes. So you can either print it and bring it in class, or you can email it to me. Okay? Awesome. So we left off talking about this sort of general process of inflammation and going through this general set of steps of inflammation, sometimes known as acute inflammation. And as I will say even a little more about, remember that by and large, in this example, you saw that the microbe was a bacteria, and the end result was that we brought in a bunch of neutrophils that did a bunch of phagocytosis to get rid of that bacteria. If, however, we were thinking about some other types of pathogens, sometimes we see different processes going on. And so specifically, I want to talk a little bit about sort of a couple pieces of what happens with viruses and what we might want to think about in terms of defending against viruses. So here you're seeing 2 different views of a viral infectious cycle, which means you're seeing basically a virus infecting an individual cell and reproducing in that individual cell. And you can see at the beginning, the first part is that the virus somehow has to get into the cell. Actually, how the virus gets into the cell is a big deal in virology. So if you think about it, phagocytosis, where you're taking something into a cell, is kind of doing the virus's job for it. Or, in some ways, phagocytosis might not be the number one best way to deal with a virus, because the virus is like, I want to get into your cell. And you're like, hey, let me let you in. Not really the most useful. So phagocytosis, yes, you can sometimes be destroying the virus, but it also, in some situations, is actually beneficial to the virus. Then you can see all of these other steps that are happening in the infectious cycle, with things going on with nucleic acids, and some translation and all of those types of things. The thing that I want you to notice about all of these steps in both of these images is that all of those steps are taking place inside of one of your cells and are really largely involving a lot of stuff going on with viral nucleic acid. So maybe you could phagocytose that viral particle outside of cells, but most of the steps of virus replication are happening inside of one of your cells. So you're not going to have a, kidney cell or a liver cell infected with hepatitis virus and then come eat the liver cell. That would be like you phagocytosing one of your cells that was problematic. Phagocytosis is, again, not really going to work here when we're looking at a whole big one of your cells and not a little tiny bacterial cell. And so we're going to need a different kind of defense that's really going to help us deal with viruses. And so we can sort of think about, if we thought about the amount of time in virus replication that viruses are inside versus outside of cells, they're mostly inside of your cells. Complement's not going to get to them. Any kind of extracellular antimicrobial peptide's not going to get to them because they're in your cell. Phagocytosis might not be a great way of dealing with them. And there's one other thing that you need to generally know about virus infection. I'll be saying a bunch of other things about virus infection in another point later on. One thing you can also notice here and here that will be really more important later is that most of the events of virus infection and virus replication happen in the cytoplasm of your cells. And so later on, we're going to be talking about a process, and we're going to be talking about different locations in the cell, and viruses are always going to be our big cytoplasm example. So you can see this is happening in the cytoplasm. And you can also see that a lot of the events involve sort of nucleic acid. And so basically what happens is we have our viral particle, and some of the early steps in virus replication involve getting to free nucleic acid. Once we have that free nucleic acid in one of our cells, so that hepatitis B nucleic acid in the liver cell, there's not really a way to get it out. We can't go in with little tweezers and be like, I want this RNA to go away, and leave all the rest of them behind. So by and large, once we have a cell that is infected with a virus, that cell is a danger to the rest of the body, because it can now be a factory to make lots of new virus particles. And there's not really much we can do to save that cell, especially when it sort of goes through this whole process of making new viruses. We can't pluck out the virus stuff. Really, the only thing that we can do if the cell gets this far into having a virus replicating inside of it is kill it, so that it doesn't form a threat to other cells by making more viruses. So we either have to kind of make it so this whole process can't get completed in that cell, or we got to kill the cell. And those are our options. This sort of side of thinking about inflammation was first really understood in the 1950s by some scientists named Isaacson Lindemann. And what they found was that if they, in a dish, they infected some cells with a virus, those cells started to make some product. And so they made a product that they secreted into their media that stopped other cells from being able to be infected. So the idea is if we have a cell that's infected, it's like, hey guys, I'm a goner, but let me give you some info so you can block this from happening to yourself. And so there's this product that was being made by the infected cells that could block infection in neighboring cells. It interfered with infection of neighboring cells, and since they didn't really know what was up or what to say about it, they called it interferon. And interferon is a type of cytokine. There are a bunch of types of interferon. Most broadly, we think about type 1, type 2, and type 3. Here, when I'm talking about innate immune responses, I'm really talking about type 1 interferon. If I'm being completely truthful, I'm actually talking about type 1 and type 3, but I know like this much about type 3. Type 3 is much newer, and so I'm really not going to mention it as much. I'm mostly going to mention type 1. And type 1 interferon is also sort of this combo of these two called interferon alpha and interferon beta. A lot of times I think a lot about interferon beta because it's the one that I think about most in my lab, but really if we can think of the type 1 interferons, type 1 interferon alpha, interferon beta, all three of those things, as far as you're concerned, can be interchangeable words. Yes, there is this interferon lambda, type 3. It seems pretty similar to the type 1s, except that it's made by barrier organs. And again, I'm not going to say a ton about it. There is also an interferon called type 2 interferon or interferon gamma. It's an adaptive immunity thing. We'll care about that later. We don't care now. The thing that we really care about now is type 1 interferon or interferon alpha and interferon beta. And interferon alpha and interferon beta are made after viral infection in particular, and they're made rapidly. So within hours, a viral infected cell is going to make interferon. In a perfect world, you can see that that type 1 interferon production is declining by like 10 hours. So this is a really, really early response in a viral infected cell. You can also notice when I look at my different types of interferons, it says that interferon alpha and beta are made by most, if not all, nucleated cells. So what does that tell you is the one kind of cell that doesn't make it? Red blood cells, because they didn't have a nucleus. But pretty much everybody else can make these. Interferon gamma is a different business with adaptive immunity. So we've got this product that's made by our viral infected cell, and it's made really early, in sort of this big burst early, and then ideally goes away. This product that's being made by our infected cell, you can see here's the virus. It's coming into this cell. It's infecting this cell. And now the cell is going to make this product, interferon. Interferon can act on a neighboring cell. Yes, this also shows it acting on the same cell. I'm not going to deal with that right now. But it acts on a neighboring cell and makes it so that that neighboring cell is better able to fight off viral infection. So now that neighboring cell is going to be in a state where it can stop having viral infection. And so now this cell, the adjacent uninfected cell, is going to be able to stop having or not have viral replication. And so this is sort of a way that I often think about this. We've got our infected cell with viral replication. That infected cell, you can see it's still infected, it's got the virus in it, makes this product, interferon. That interferon, and specifically type 1 interferon, that type 1 interferon binds to the type 1 interferon receptor on a neighboring uninfected cell. And this neighboring uninfected cell now can block viral replication and is not going to allow the virus to replicate. It's known as the antiviral. So we refer to this cell as now being in something called the antiviral state. So it's very similar to what you saw with acute inflammation. I'm coming. This is very similar to what we saw with acute inflammation. Before we were seeing inflammatory cytokines like IL-1 beta, IL-6, TNF alpha. Here it's a different cytokine. It's type 1 interferon, with the different pathogens. And instead of getting to phagocytosis, we're getting to the antiviral state where we're blocking the ability of this cell to allow viral infection. So this is sort of the big picture idea of what goes on of this process. Hopefully you see the ways that it's similar to what we've seen before and some things that are a little bit different about it. Yes, Shelby. It's okay.
Does this just prevent infection, or does it prevent just the replication? Does it stop it from getting infected, or does it stop the replication if it does get infected?
That is a great question. We're coming to that. Okay. There's a slide that I want to get to, and then we can sort of address that, okay? And so this is sort of showing you a lot of the same kind of information. Here's my infected cell. See, there's a virus, and it's making interferon, and it's a different cell because I made a line. And that interferon is binding to the interferon receptor, and you can see that things are happening in this neighboring cell. When type 1 interferon binds to its receptor, there are lots of new proteins that are made in this uninfected cell. So this is starting to get to your question. This cell is going to make a whole bunch of new proteins. Those proteins are called ISGs, or interferon-stimulated genes. So if you stimulate with interferon, you get interferon-stimulated genes. Again, not very complicated or clever with our naming here. There are thousands of them. The combo, it seems like with different viruses, like you get a different combo of ISGs. That's probably really important. I'm just going to say ISGs are the things that are made from interferon and get at that. But actually, if you get into this, it is a lot more complex. So you can see the ISG combination is probably really important. Most of those ISGs all have a really similar promoter that allow them to get turned on downstream of interferon signaling. That promoter is known as the ISRE. As I mentioned, these ISGs are really what allows our cell to either block infection or to block replication, coming back sort of to your question. And to best kind of think about this, we need to think a little bit about, okay, so how do they do that? How do these ISGs work to block infection? Now remember, the ISGs are made in the neighboring uninfected cell and are helping that neighboring uninfected cell block further rounds of viral replication. And so what I want to do is I want to tell you about a few examples of ISGs and how they work. And I think that that's going to help us understand the antiviral state a little bit better. So these are some examples of some ISGs that are made during the antiviral state. So one of them is a gene called PKR. One of them is a gene 2'5' oligoasynthetase, which eventually leads to this thing called RNA cell, MX proteins, IFIT proteins. Like I said, there's thousands of them. I could list thousands and thousands. But I want you to see what some of these examples do so we can think about what's going on with the antiviral state. So when protein kinase R is activated, it eventually leads to activation of this other protein, EIF2 alpha, and the overall result is inhibition of translation. And so the idea is, if you think back to that process of virus replication in the cell, at some point we have that viral nucleic acid in the cell and we need to do some translation from mRNA to make new virus proteins. And so now it's really a question of the replication cycle. Virus got into the cell, it started to do its steps, but then when it needed to make proteins, we inhibited protein making. So it couldn't get all the way through the cycle. So in this example, it couldn't get all the way through the cycle. I think every example I'm going to tell you about is going to be you can't get all the way through the cycle, but I also think, there are some examples that would actually be block infection examples. So my answer to you is it depends on which ISG we're talking about. So here's another one that says inhibiting translation. The other one I'll mention here, you can see 2'5' oligoasynthetase, eventually leads to a product, RNase L, that results in mRNA degradation. So our virus, is going to release nucleic acid. We're going to get to mRNA, and then we're going to have this enzyme that's going to cut up the mRNA. So again, the virus can't complete a full round of replication. And I could go on and on and on with all sorts of super exciting ISGs. But I also want you to look at this and think about it in another way. So let's imagine, say, protein kinase R getting turned on in your cell, or 2', 5' OAS. We're going to inhibit translation or degrade mRNAs. There's one thing that's actually not indicated on this slide that you might not notice is missing. When it says inhibition of translation, it doesn't say inhibition of translation of virus proteins. It says inhibition of translation. We stop translation in the cell. So we stop making virus proteins, and that foils the virus. But we also stop translating all the other proteins in the cell. Here, we're going to degrade mRNAs, and yeah, that's going to foil the virus because we're going to degrade the virus's mRNAs, but we're going to degrade all the other mRNAs in the cell too. Does this sound like a great scenario for the cell? No. And I think that's really important to mention because sometimes when people hear about sort of this process, interferons inducing the antiviral state, they say, I don't like this. I would like my cells to be in the antiviral state 24/7. Why are my cells not, why can they sometimes be infected? If they have a way to repel infection, can't they just do that all the time and then I'm never infected and I live happily ever after? And so what I want you to see is that the antiviral state is not like super awesome healthy cell time. The antiviral state is a pretty stressful, pretty difficult situation for the cell. Ideally, if the cell doesn't do translation for 12 hours, it might be able to live through that, and it might be able to, not translate viral stuff. Viral stuff might get degraded, and then the cell can kind of go back to its normal life. But that cell's going to be in bad shape for those 12 hours. And you can imagine the same thing from some of these other processes getting blocked. And so what I want you to realize is the antiviral state, again, not a totally healthy state for the cell because a lot of cell processes can't happen in the antiviral state, but it does have the benefit of blocking potential virus replication. And so, one of the other textbooks I have refers to the antiviral state as, or makes an analogy to an antiviral state being like stopping a forest fire by cutting down part of the forest. So that this fire can't spread and you end up protecting the part of the forest that you didn't cut. Like if you put a little cut area. No, nobody wants to cut down the forest. But if you save the whole organism by having a few cells that are uncomfortable for a few hours, that's actually pretty good for the organism. And so I mentioned, with the type 1 interferons, that type 1 interferons can be made by pretty much all nucleated cells. And so I often like to think about type 1 interferon as sort of a message to say, hey friends, I'm done for, but you guys protect yourself, right? The type 1 interferon is the call coming from the first cell, and that first cell could be pretty much every nucleated cell. What cell would you expect should have an interferon receptor, should be able to listen for that call? Yeah, Jonah. You would expect basically every cell is going to have an interferon receptor. So interferon makes gene products that are not awesome for our cells, especially if we had them for long periods of time. And most of our cells have receptors for interferon. So if you start making interferon, particularly if you start making a lot of interferon, you're going to have changes to your physiology. You are going to know it. And some of those changes to your physiology that you're going to have are going to be fever, chills, nausea, and malaise. You're probably only going to have them for a short period of time because that interferon is only made for a short period of time, in the first few hours after infection, but you're going to see those kind of symptoms. And basically every viral infection leads to an interferon burst. A lot of people refer to having fever, chills, nausea, and malaise as flu-like symptoms. That's why every virus pretty much gives you those symptoms. It's because every virus is leading to an interferon burst. And so sometimes people will be like, I have the flu. And I'll be like, you have a virus. You have interferon in your body. I don't know if you have the flu. We can do specific testing to look for that specific virus, but most likely it's just you have some amount of interferon going on. One other thing that I have on the right-hand side of this slide, which I will also kind of say a little bit, teach a bit more about going forward, is that we talked about sort of the innate immune response as like two different parts. There's this type 1 interferon part and this acute inflammation, TNF, IL and beta IL-6 part. Those two can interact with each other. And specifically, we can see things like interferon turning off the acute inflammation phase. It's pretty important that your immune system sort of cross-regulates itself in this way to stop pathology. If all the immune responses were happening all the time, you'd get a lot of tissue damage. So you kind of do one and tell the other ones to shut off. But it also means that if you have, say, a lot of interferon being produced, you're actually suppressing other parts of your immune system. Many people who die of influenza infection actually don't die of influenza infection. They die of a second bacterial infection that they got afterwards because the interferon that was helping them fight the flu turned off their antibacterial response. And they end up dying of a bacterial infection. And so sort of there's this on and off between some of these responses. I have already said this a couple times, but I'll just try to make it clear here. This is a way that I often simplify what we've talked about with cellular parts of innate immunity. So you can see it says very oversimplified overview. There are many ways immunologists who like professional immunologists would hate what I'm going to say, but there are also some reasons why I say it. So like they can deal. So remember that in the top, we had a pathogen that was bacteria. And in the bottom, we had a pathogen that was a virus. And then, see, I wrote the pathogen thing still. Pathogen, bacteria, pathogen, virus. At the top, we saw cytokines that were made that were sort of these typical inflammatory cytokines, TNF alpha, IL and beta, and IL-6. In the bottom, we saw those typical cytokines being the type 1 interferons, which is interferon alpha and interferon beta. So you can see kind of similar processes, different official players. And you can also sort of compare at the top, a lot of what we were trying to do was phagocytosis related. We were trying to recruit phagocytic cells. We were trying to improve phagocytosis with this process. At the bottom, we're trying to get the antiviral state. And so this is a little bit of our compare and contrast. What you can might also be able to determine from all of this is that, or what I can tell you is that in both cases, we had some kind of receptor, some kind of pattern recognition receptor that recognized the microbe. There was one that recognized bacteria and said, aha, this is bacteria. I should make TNF, IL and beta, IL-6 and do the phagocytosis route. Or we had a receptor that said, aha, this is a virus. I should make interferon and go this antiviral state route. And so, in fact, this differentiation started at the level of the receptor that first saw that individual pathogen. And when that receptor led to signaling. In both cases, in our infected cell, we saw transcription of new genes that we weren't previously transcribing, right? We said, okay, now we're going to make, in the top case, the TNF-alpha mRNA, so we can make TNF-alpha protein. And we're going to make the IL-6 RNA, so we can make IL-6 protein. And we're going to make the IL-1 beta RNA, so we can make IL-1 beta protein. Versus, oh my gosh, we're going to make interferon alpha RNA and interferon beta RNA, right? We're basically doing different amounts, different transcription. We can also divide up a lot of these processes and these PRR signaling pathways related to that transcription, because we find that there are two big transcription factors that tend to be associated with PRR signaling. So one of the transcription factors is a transcription factor called NF-kappa B. For reasons that I sort of understand. Students always want to call NF-kappa B NF-kappa beta, but if I told you all of what NF-kappa B stands for, the B stands for B cells, so it's a B. And if we have a receptor that eventually tells us let's turn on NF-kappa B signaling, we get, oh my gosh, those inflammatory cytokines, the same ones I listed previously, TNF, IL-1, IL-6, and some chemokines and some other things where we eventually get this sort of acute inflammation. And so that top set of processes I was telling you about on the previous slide are really all sort of the results of our macrophage turning on this transcription factor, NF-kappa B. While if we are going to see type 1 interferons getting made in order to lead to the antiviral state, we're going to have a type of transcription factor called IRF. IRF is what makes interferon. IRF stands for interferon regulatory factor because it makes the interferon. And so that bottom pathway that I was telling you about before is largely because the infected cell somehow gets a trigger to tell it to make this transcription factor, IRF. This is all the very end of that PRR signaling process, which I showed you before as sort of like a magic arrow. And one of the things I wanted to spend a bunch of time on today is telling you about some of the actual PRRs, some of these receptors, and what we know about them and what they bind to. One of the big things that I'm going to be telling you about when I tell you about these groups of PRRs is I'm going to be like, this kind of PRR leads to NF-kappa B. This kind of PRR leads to IRF. And so you're going to see it tie back to that transcription that I was just telling you, the transcription factors I was just telling you about. But I also have one other slide that I added in before this one, and I will be perfectly honest with you, I struggled a lot with where the heck should I add that slide. So as I'm talking right now, it feels completely awkward and like not the right spot, but I also kind of don't know where else to put it, so we're just going to go with now. And it is somewhat related to your assignment that's due on Friday. And what I just wanted to sort of mention is that we now understand that all of these processes where we make innate immune cytokines are super important for directing your adaptive immune response. Adaptive immunity doesn't work right if these innate immune processes don't work right. I first learned about this from reading about a scientist named Janeway. I have a Janeway figure on your left. It's in one of those readings. The like optional readings is actually this Janeway paper. And there was a lot of controversy in the field, an argument between Janeway and another scientist named Matzinger, so I'm giving them both credit here, because it's a whole thing, and I just don't want to go into it. But they basically sort of came up with this idea where they're like, wait a minute, an adaptive immune cell doesn't just work. Like if your T cells and B cells just see an antigen, the thing that they see, they actually usually don't get turned on. They only get turned on if they see the antigen and some stuff from the adaptive immune system. So adaptive immune system only, nothing. Antigen only, nothing. Only when you get sort of this combo of an adaptive immune signal from a macrophage and sort of the classic signal that everybody thought of. You can see this over here too, that here is my T cell. My T cell is responding to something, and it doesn't actually get turned on. It might actually get killed. But if that T cell now responds to that while also responding to adaptive immune products, we're going to actually have that T cell work really well. Matzinger talked about this. Matzinger referred to this as sort of the danger hypothesis or the danger signal. So the idea is, if one of your T cells sees something and itself, one of your T cells sees something and it's not actually harmful, you don't want the T cell to respond. But if the innate immune system has seen something and is like, bad, bad, danger, danger, alarm bells, alarm bells. And so now the T cell sees, okay, the innate immune system is doing alarm bells and I see something. This thing must be bad. And so it's a great way to kind of avoid things like autoimmunity, because your response to your skin cells shouldn't have any danger around. So you should be like, nothing. But if you have this additional danger, that is going to really lead to your adaptive immune response being fully activated. We're going to see this a lot more when we talk about particularly T cell activation. But part of your assignment was sort of, and part of the reason why people were so excited about thinking about PRRs and thinking about the Nobel Prize was realizing that understanding this process actually was understanding in all these inflammatory diseases and how you control the adaptive immune system. And it was like kind of a big thing to understand. So this is sort of basically, like I said, there was controversy here with Janeway and Matzinger. There is also controversy in terms of who proved this. This was just a hypothesis these people had. And whether the right person got the Nobel Prize for proving it or whether the wrong person got the Nobel Prize for proving it. So that's just something to mention. All right, so I'm going to go into thinking about the groups of PRRs. This table shows 5 different groups. I'm going to talk about some of them. But the first ones that I want, one thing that you can notice is if you look at all of the groups, they usually have LR in their name. It's something like receptor, at least originally. And the first ones I want to tell you about are a group that are called toll-like receptors. Because toll-like receptors were really the first ones that were described. They are the ones that are subject to that argument in the papers you're reading about of who proved it first. They kind of began this whole field. To understand toll-like receptors, I have to tell you about a protein called toll. Because there's this protein called toll that was described, and then people eventually said, oh my gosh, there are other things like toll in humans, so we called them toll-like receptors. And the person who discovered toll is a woman named Christiane Nilsson-Volhart. She also won the Nobel Prize, but she was not an immunologist, she was a developmental biologist. And she first was trying to understand a process called dorsoventral patterning. So basically in an embryo, how an embryo gets to have a front and a back instead of all being the same. Sort of, how you get sort of head versus tail and front versus back is what she was trying to do. That was what she was trying to, and she was looking at flies and looking at fly embryos. And she was trying to mutate genes and try to figure out, like, do some of them make the embryo completely develop wrong? Like, and not have a front and a back. So one day, she had these fly embryos. She made a bunch of mutations, and she just was looking at fly embryos under a microscope and, like, hoping she'd see some that didn't have front and back. And while she's looking through her microscope, she said, "Toll." She's German. Brenna. What does it mean that she said toll?
I mean, I thought it was supposed to be like great, but I'm assuming that case is like, oh my god, or like.
Yeah. So when I first learned this, I learned that it was holy shit. But it's some kind of like explanation of like, oh my gosh, she found the messed up embryo. And so the gene that was mutated in that embryo got named toll. It turned out that you could actually do some fancy genetics and get flies that had mutations in toll to grow up normally, but then you could just see what that gene was doing in the adult. So basically, imagine development fine, but then you turn off the gene as an adult to see is the gene doing anything as an adult. And when they did that, this is the image of what they found, was that the flies all died. All of this, like, up here and all of this is fungus. This entire fly is like super, super colonized by fungus, and all the flies died of fungal infection. And they said, not only does toll have a role in development, toll seems to also have a role in the immune responses in flies. And they eventually realized that it was the first pattern recognition receptor. It was recognizing that microbe and leading to an innate immune response in the fly. And others found that there were similar receptors, structurally similar, really similar receptors in humans and in many other organisms. And so they called them the toll-like receptors, or the TLRs. You can see the structure of TLRs here. You can notice that they have sort of this one domain that allows them to bind to microbes. Officially, that domain is called an LRR domain, and they also have a domain that's involved in signal transduction. LRR, leucine-rich repeat. We now know in humans that there are about, I think we're currently talking about 13 toll-like receptors in human. Though, you know, we're still working on discovering this. Oops. When we think about all of those toll-like receptors, or TLRs, we find that they actually fall into two groups. One group hangs out on the membrane of cells, particularly on the membrane of macrophages. So these TLRs are looking for things that are outside of macrophage, or whatever cell. There's another group that do surveillance inside of cells, specifically inside of a cell compartment called an endosome. So we've got TLRs that are on the surface. We've got TLRs that are on the endosome. And if we look at these, you can see that actually, if we look at the genes in humans and how they've evolved, they really evolved as two separate little groups. And one group, the ones that are on the outside of the cell, tends to bind to types of lipids. The ones that are on the inside of the cell tends to bind to nucleic acids. The most famous lipid that's bound by one of these cell surface TLRs is a lipid called lipopolysaccharide. Some of you have heard of lipopolysaccharide. What is lipopolysaccharide, or LPS? Yes, Jonah. All bacteria? Gram-negative. So LPS is a note L. The L stands for lipo. It's a lipid. And it's on the surface of certain types of bacteria. So if you had to guess, don't overthink this. Well, no, I won't make you guess it. I'll just tell you, but because it's all kind of straightforward. Like I said, we've got two groups of TLRs. One of the other big things between these two groups of TLRs is what happens if a signal goes through that TLR. So what happens as a result of signaling through either of these groups? We got two groups, right? And I told you before we got two transcription factors. Those numbers match rather nicely. The TLRs that are on the surface of the cell are usually binding to things from bacteria, like LPS, like those lipids. And those cell surface TLRs all induce NF-kappa B signaling. And so they're part of that sort of acute inflammation, like making IL-6, IL-1 beta, TNF alpha. Do you need to know all of the different players between a cell surface TLR and NF-kappa B? Nope. I'm just telling you it's a thing. Alternatively, we've got those endosomal TLRs. And remember, they're binding to nucleic acid. What kind of pathogen might be particularly one that we want to detect with nucleic acid? Viruses. The endosomal TLRs all signal through IRF. And so generally these are looking at nucleic acids. Those nucleic acids are usually from the virus. Oh my gosh, we're going to turn on the transcription factor that gives us the good virus response. And so what I want you to see for these PRRs is that there's a bit of a sort of correlation between what's the pathogen, we respond to what kind of pathogen we have, and we make the right kind of response for that pathogen. And we kind of set up the immune response to go in a direction we want. I have absolutely no idea why the next two slides are there because they're duplicates of these two. I got nothing. I know. Who was the amateur who was putting these together? So that's what I wanted to tell you about TLRs. I'm not going to say a ton about the CLRs, but I do want to mention a little bit about some of these other ones. So one that I want to mention are the RLRs. And so the RLRs, notice what's cool. One of the things that's cool about the RLRs is that the RLRs are really looking for their microbes in the cytosol. So the TLRs were looking on the plasma membrane and the endosome. The RLRs are looking in the cytosol. So we're doing surveillance of lots of different places. We're doing on the lookout for microbes in all sorts of different places. Because the RLRs are doing surveillance in the cytosol, what microbe do you think they might be related to? Viruses. So they're largely going to be looking for viruses. So these receptors, the first one that was described is called RIG-I. So they're RLRs because they're RIG-I-like receptors. Are found in the cytoplasm and are going to bind to viral nucleic acid in the cytoplasm, and then they're going to turn on our transcription factor IRF. So again, we're pairing the location in the cell and the type of pathogen with an appropriate type of response. What's up? Hey. Okay. No, he just writes stuff. I did. Yes. So the next group that I want to mention is a group known as the NLRs. The NLRs are tricky. The top of this slide tells you what NLR stands for. And what you see is that it does not stand for N-like receptor, right? The reason for that is that it used to stand for N-like receptor, and then we learned that the protein that was N is actually super weird, and the other ones are not like it at all. But we liked the name NLR. So we tried to make up a new name that was true that also had NLR. And you can see it's convoluted to try to make, they had to work to try to make that happen. So they officially are the nucleotide binding domain leucine-rich repeat proteins. But one thing to note is they do have this region, the leucine-rich repeat, which, have you heard of that before? Brenna, have you heard of it before?
You're supposed to? It's not coming to me.
It was that LRR that you asked about in the toll-like receptor, that domain that you were writing about. So in fact, they've got some structural similarities to the other ones. They still have the same kind of structural areas. They have this middle domain that binds to, that's a nucleotide binding domain. And then they have a domain that is involved in protein-protein interactions. So you can see that structure here. And so these, at the bottom are the NLRs. We further divide them into groups based on what protein-protein interaction domain they have. So some of them have this domain called a pyrin. They end up being called NLRP. And some of them have card, and so they end up being called NLRC. It's really, real fascinating. But what I want you to notice in this figure is that the NLRs, in fact, have some similarities to a bunch of proteins, structure-wise. So you can see two other proteins that have similar structures shown at the top. One of those proteins, the one that's at the top that says NBS LRR proteins, those are the main proteins of the plant immune system. And so this exact same kind of structure, this exact same kind of process that we see with NLRs turns out to be a big part of plant immunity. So it's sort of a nice example of convergent evolution, perhaps. And then we also have APAF, which I will come back to in a second. So NLRs can do two different things. How much they do each one depends on which NLR. I don't care that you know the specifics of this NLR does this and this NLR does that. But some NLRs will bind to their microbe product and will lead to cytokine production. In particular, NF-kappa B activation and our inflammatory cytokine production. So some NLRs lead to cytokine production. Let's see. This says they're found in the cytosol. We'll go with that. But they also have this second function. And to think about the second function, it's useful to think about this similar protein called APAF1, which you can see has really, really, really similar structure to an NLR protein. It's too bad that Dr. Dunaway isn't here right now, because if he was, I could have you guys tell him if you've ever heard of APAF1 before. Because in fact, you learned about it in Bio 250. So, you may not remember APAF1 from Bio 250. But when you learned about APAF1, when you were learning about apoptosis, which you remember was a form of programmed cell death. And when you learned about apoptosis, you learned about a bunch of proteins coming together to make this structure that allowed for caspases to be activated. That structure was called the apoptosome. The apoptosome, the structure that was made in apoptosis, right? Dr. Dunaway loves to call this structure the pinwheel of death. The pinwheel of death might be the thing you remember of all the things. So APAF1 gets activated when the cell wants to go to apoptosis. It comes together in this big structure, and because it's a structure that's made during apoptosis, we call it the apoptosome. Turns on some caspases, gives you apoptosis. NLRs have this really similar structure, and many NLRs can also make a pinwheel structure. But that pinwheel structure is not meant for apoptosis. So it doesn't get to be called the apoptosome. Instead, this pinwheel structure is made for inflammation. And so what would you call a pinwheel structure made for inflammation? Yeah. You call it the inflammasome. And so NLRs also have this second function where they can come together to make this complex that includes multiple NLRs and some other additional proteins and make an inflammasome. The inflammasome leads to some caspases getting active. The apoptosome did too. It led to caspase 3 getting active and you getting apoptosis. This one is really going to lead to caspase 1 getting active, and caspase 1 helps us make inflammatory cytokines. So it's very, very similar. And this is one figure from your textbook trying to show the inflammasome being made by some of our NLRs. Here you can see sort of another version of it, that we have our bacteria. It will get sensed by sensors that are actually not shown there very well, and we'll turn on these caspases and we'll turn on downstream processes because of the inflammasome, my lovely little pinwheel. The inflammasome is interesting because it involves helping with inflammatory cytokine production, but it also is involved in a second thing. When we saw the apoptosome, you remember the apoptosome was involved in the programmed cell death form apoptosis. A lot of times when people come out of Bio 250, they have the idea that the apoptosis is the only form of programmed cell death. You either die by mistake or you die by apoptosis. It's actually not true. There's so many forms of regulated cell death. I can't even keep up with how many we have now. I, in fact, told you about netosis with neutrophils. Technically, that's a programmed cell death form of neutrophil. And you can see apoptosis on the left. Remember, with apoptosis, we get sort of the cell breaking into these little blebs, these little membrane blebs of pieces. It's sort of like the cell doesn't want any of its toxic stuff to go anywhere, so it's gonna shelter it and make little things. And it's gonna doesn't want to bother anyone around it. I think of apoptosis as very quiet cell death. I'm just gonna die right here by myself and not bother anyone. It's kind of how I think of apoptosis. There are other forms of cell death where the cell is going to sort of explode. And one of them that we've learned a lot about is this type of programmed cell death called pyroptosis. So instead of apop at the beginning, which is falling, we have pyro. What's pyro like? Fire. Pyroptosis is fiery cell death. Or I often think of it as loud cell death. With pyroptosis, we're not going to try to constrain any of our stuff. We're not going to try to be quiet and not disturb our neighbors. We are going to throw everything out of our cell and be as loud as possible and disturb all of our neighbors, because that might give them a signal that they should protect themselves. That might tell them something bad's going on in the neighborhood. And they should deal with it. And so with pyroptosis, we have the cell swells up, the cell ruptures, the cell loses all of its intracellular components. Those might be things that further inflame the area, and so we get this inflammatory cell death. And the inflammasome also induces pyroptosis to happen. And so here you can see a cell just exploding and releasing all of its stuff as it's undergoing pyroptosis after inflammasome formation. So just 'cause I can, these are some of the things that have been shown to induce NLRs to make an inflammasome. You can see that we've got things like ATP, cholesterol, urate, which is found in joints of people with a disease called gout, amyloid beta, excess glucose, asbestos, silica, UV radiation, bacterial molecules, viral molecules, fungal molecules, protist molecules. So you can see there are a lot of things that seem to induce this inflammation and induce this kind of inflammatory response. And exactly how some of those structures get bound is a whole other story, a whole other time, but it's pretty interesting. There are a number of diseases that are shown to be related to having a mutation in one of your inflammasome-related proteins. And so these are basically diseases where you have like random fevers and random inflammation for no reason. One of them is, so you can see there's like familial Mediterranean fever or pirate-associated autoinflammation or things like that. There's actually one where you basically get inflammation from being in a hot shower. Like it's all sorts of things where your NLRs are actually responding inappropriately and triggering inflammation and pyroptosis very frequently. And so there are a lot of disease states that are related to these. This is actually a super out of date table of some diseases that have specifically been shown to be related to the inflammasome. Either because of genetic mutations, the way that they respond to certain drugs or animal models. And so you can see we've got some of those autoinflammatory diseases here, but you can also see inflammatory bowel disease, type 2 diabetes, hypertension, Alzheimer's, atherosclerosis, dermatitis, type 1 diabetes, multiple sclerosis, lupus, vitiligo, Addison's disease, celiac, rheumatoid arthritis. Basically, like, if you can think of it, it's probably related to the inflammasome. I went down a big time rabbit hole when I was putting all this together. So I'll just also tell you, for example, diet seems to dramatically impact inflammasome. And so remember, both some of the NLRs and TLRs, remember TLR was binding to lipopolysaccharide, which was a lipid. It seems like diet is influencing basal inflammation levels pretty dramatically. And so here they're saying, this healthy diet means that you have low levels of systemic inflammation. And if you eat this terrible Western diet, then you have high levels of systemic inflammation, and it's all due to signaling through the inflammasome. And again, I tried to to make a slide with one figure on it and the level of rabbit hole I went down. But these are some of the things I read. There's one other group of PRRs. This group of PRRs is listed here as the ALRs. I'm not going to have time to jump way into them, which is really sad because they're the ones I work on. But what I want you to notice that's really key about the ones that are left are that they're in the cytosol and nucleus. So there is no part of your cell that goes without surveillance. They are largely looking at nucleic acid, particularly DNA, and leading to that interferon response. They have lots of really cool features, some of these DNA sensors are actually conserved all the way back to bacteria. So bacteria have them, and that's how bacteria can help defend themselves against bacteriophage. It's all super fabulous. If you want to know more about it, I could tell you about it forever because that's what I work on in my lab. But on Wednesday, I guess I have to start adaptive immunity. I guess I have more geeking out about innate immunity slides, but I think I have to hold myself back. So I think on Wednesday we'll start with adaptive immunity, and I will see you guys then.